You are listening Audio Paper Series with Yasir, and today's guest is Dr. Waters from Quorum Institute, Norwich, UK, to discuss one of her publications, Impact of Salmonella Genome Rearrangement on Gene Expression. It is published in Evolution Letters. Welcome, Emma.
Hi, Yasir.
Thank you for joining us. What is Salmonella genome rearrangement?
So genome rearrangement is where large genome fragments shift position and orientation in the genome to ultimately produce just different unique genome structures. And this doesn't actually necessarily affect the underlying nucleotide sequence. So these large structural variations occur via homologous recombination around long repeat sequences, which in the case we are looking at here are the ribosomal operons, which are present in all bacteria. Now in Salmonella, there are 7 ribosomal operons, which separate the genome into 7 fragments. And because these ribosomal operons are so similar, homologous recombination can make genome fragments lying in between them flip orientation or move somewhere else in the genome.
So, is this a continuous process or is it a rare happening?
This is something we don't actually know right now. So we believe it's something that can happen and then it may revert or it may continuously evolve into something else.
How did you investigate it as gene rearrangement is happening? What kind of methods did you use?
So historically, the identification of genome rearrangements was challenging and it was performed on an ad hoc basis with low resolution methods such as long-range PCR. Now, this was the method that was originally used in this work, which was started by Jana, a master's student all the way back in 2008. Jana designed forward and reverse primers to bind to regions which are a few hundred base pairs downstream or upstream of each of the 7 operons. Now, these primers were used to perform 91 individual long-range PCRs to test all the combinations of the neighbouring fragments. Now, 91 is a minimum, so poor Jana, just for one isolate, she had to do these 91 PCRs continuously. And in the end, in her year, she got 5 isolates with different GSs. In this work, we present the first use of long-range sequencing for our oxygen nanopore MinION to confirm genome structures identified by Jana and hopefully to replace this original benchmark method.
So in past, long-range PCR was being used, but in this study, you have used nanopore long-read sequencing.
Yeah, so in this particular one, we've used both just to show that you can replace the benchmark method.
Perfect.
And with long-read sequencing, you can actually gain more information. So you gain all the genetic information that normally comes with sequencing, and you can actually do this in a higher throughput.
So would you like to share what is your major finding in this study?
Sure. So in this study, we look at 4 rearrangements, and we investigated these with differential gene expression using transcriptomics. Isolates which generally had changes in genome rearrangement relative to the parent strain were accompanied by changes in the gene expression, and rearrangements with similar fragment movements demonstrated similar changes in gene expression. Now, we had one really extreme rearrangement which caused a large imbalance between the origin and terminus of replication, which is a major contrast to the parent strain, which had a balanced genome, which means that the chromosome replicates, and as it does so, it does it by 2 strands of the same length, starting at the origin and resolving at the terminus at roughly the same time. But in this extreme imbalanced genome, this had a direct impact on replication and growth rate of this isolate, as there is an extended replicor which takes longer to be processed. Now, this sounds disadvantageous to the bacteria, but also has a strong knock-on effect on the gene dosage, which is where genes are now close to the origin of replication have a higher copy number and genes are now further away.
Very interesting. So Salmonella can modulate gene expression by genome rearrangement. Do you think that Salmonella genome rearrangement is part of its evolutionary process?
We definitely think so.
Thank you, Emma, for sharing very interesting science.